All right. For today, go ahead really quick and I want you to go to this link and answer these questions. There's just two and they're very short. So if you have the PowerPoint pulled up on Blackboard, you can actually just click on the link, which is a cool feature. You could type this in, I guess, if you wanted, but it should take you to a Google form. So go fill out my poll, please. No, you would have to just click on the PowerPoint if you wanted to click on the link, which is posted in the folder. And when you're answering the question about proteins, try not to use any resources. I'm just truly curious what you all are going to answer that question. So if you go to the lecture PowerPoints, you can click on today's lecture and you can click on the link or copy the link from that. How are we doing? Almost done? All right. Do you need more time? Okay, we're gonna get going then. Couple reminders for you. Next Friday, there's no class. I'm not going to be here, so you do not have to come. There's not going to be a recording or an activity or anything. We just don't have class that day. I do also want to remind you, exam one is 2 weeks from today. So it is, again, going to be broken into two parts. We'll have an in-person short answer on paper version, and then there will be a take-home version, which is just take home's the wrong word. It'll be online during class or anytime at that day. That'll be short answer. So it'll be kind of two-part short answer and multiple choice. Yeah, so on Friday, February 14th, you'll come to class for the in-person short answer version, and I'll have like printout version for you. Then the following class period, I'll give you off, and you can use that class period to do the multiple choice. Yep, it would be the following Monday. Yep, so I'll give you 2 days. It's not gonna take you probably that whole time, but whenever I do like online assignments, I try to give you time back so you don't have to come to class during those times. Mm-hmm. Yep, but it's not like two full exams. Like the short answer would be 10 questions maybe, and then the online one will be like 15 multiple choice questions. So it's not like 300 questions, like a crazy amount of work. It'll be about an hour. It should take you about an hour to do probably both parts.
You like the
I do have exam review built in. Yep. the day before. So it would be Wednesday the 12th, where I can give you more specific details on like number of questions and time limits, because they don't have it fully written at all right now. So there's still some things that, like the number of questions that could change. Anything else? Okay. So we're going to talk about this image. I actually have kind of a fun video as promised. I'm going to have you answer this question, but I have it on the slide after. So this is a real picture, kind of cool. This is the basilisk lizard. They tend to live in the neotropics. And hopefully I have a video that I will play for you. And the sound is kind of silly. It's kind of funny. So hopefully we can get that to work.
Why play like everyone else? With Maverick Rewards at Hoot Hill Casino and Resort, designed for players who think and play outside the box. The personalized loyalty program that puts you in control.
Okay.
Costa Rican lizards are looking for food. Basilisks have been nicknamed the Jesus Christ lizard. Why? This adult male, probably the father of the young lizards, can't tell us. Nor can this female, exclusive property of the territorial male. It has nothing to do with the feeding habits of the basilisk, which consists primarily of insects and berries. The toy reptile will reveal the secret as it stalks the young basilisk. The basilisk is called the Jesus Christ lizard because it can walk and, well, really run on water. It bicycles its hind legs, and the tail becomes a counterweight. We can't know what the snake is thinking. Did it really want to eat the lizard, or did it just want to show?
Okay, so kind of silly. That video was from 12 years ago, so you can see the vintage kind of moment there. All right, I am going to pull that question back up here. So the basilisk lizards, and again, those weren't edited videos. They actually do really look like that. I have seen them in the wild, and it's very interesting. Given what we've learned about chemical bonding and molecules, how is this possible? I'd like you to think about this answer by yourself for a sec, come up with the answer, and then I'm going to have you share with a partner, and then I'm going to have you share with me. So think about this. If you have an answer, tell someone near you, and then you're going to tell me. Hey, what are y'all thinking here? How is this possible? As pertains, yeah, cool. I was going to say as pertains to bonding. But yeah, the main reason this is possible is because hydrogen bonds create surface tension on water's surface. So this next slide is kind of re-emphasizing that idea. Remember that water molecules are polar molecules, so there's an unequal distribution of charges on these molecules. Therefore, there tend to be slightly positive and slightly negative areas on these molecules. When you get lots of water molecules next to each other, we tend to see lots of hydrogen bonds, which are represented by these dotted lines in this larger figure, which is showing lots of different water molecules together. Same deal here in the slightly zoomed in figure where you can see, again, those hydrogen bonds represented by the dotted lines. So the basilisk lizard is able to take advantage of the cohesive forces of water and the surface tension. Now, this figure I did not show you last class. It's kind of interesting because it's showing like something that has mass on the surface of the water, and it's explaining the fact that we see stronger cohesive forces at the surface of the water because it's not going to be bonding with the air. So these water molecules at the top near the surface, they're not going to be extending any force into the air, just back into the water. So that's why we get that strong cohesive force at the surface of water. Questions here? Okay, I did hear some people talking a little bit about geckos. Now, geckos are another lovely lizard-based example for a different type of bond that is similar, but geckos are a good example of van der Waals interactions. So van der Waals interactions are also weak intermolecular interactions, but they are different from hydrogen bonds. So make sure you kind of separate those two things in your brain. Today we're going to talk about macromolecules. We are going to have this in two parts. So today we're going to talk about what macromolecules are, how they're formed, some structural properties of carbon, important things that explain macromolecules. And then on Monday, we're going to talk about proteins, lipids, and nucleic acids. So we have 4 categories. Today we'll be focusing also on carbohydrates. Your learning objectives are broken into 2 categories, basically, the ones we're going to be talking about today. And then the purple are what we'll talk about next time. So today we're going to talk about what macromolecules are. A really key piece here is I want you to make sure you know the different types of carbohydrates, their structure, properties, functions, and examples. So this can be challenging as I start to show you like carbon rings and carbon-based structures. Sometimes that tends to get a little bit jumbled. But I guarantee you, I will ask you to identify these structures in the future. So not super duper hard ones, but I would expect that you could differentiate between like a carbohydrate and a lipid if I just showed you 2 pictures, for example. So at the end of our macromolecules lecture, if you're struggling with that type of like visualization, it's important that you either ask for help or kind of ask for resources to study those types of differences. Because again, that's the type of question I would ask on like the written exam, like what is this structure, for example. So as I was talking about last class, when we were talking about valence shells and chemical reactivity, carbon has 4 valence electrons, which makes it very likely to form lots of different covalent bonds. It often forms 4 bonds to fill its octet, and we tend to see covalent bonds in this most often with carbon. Now, what's interesting about the type of structures that carbon tends to form, they have what we call tetrahedral geometry, which refers to the angle between the bonds, which is shown here on that figure with the red and the dash. So carbon tends to form these perfect 109.5 degree bond angles, which makes this tetrahedral arrangement. And in this figure, the dashes and the filled in lines are essentially just referring to the 3-dimensional directions of those atoms based around that central carbon. So that's what that figure is showing. You don't need to know stuff like that necessarily, but we do want to know that it has this tetrahedral geometry, which is really structurally sound. This tetrahedral form allows carbon molecules to link up with other carbon molecules and form really stable structures. So we say that carbon forms backbones, or we tend to see these carbon backbones, and those comprise almost an unlimited number of molecules. We see lots and lots and lots of different carbon-based molecules because it can form single, double, or triple covalent bonds. As I was talking about previously, carbon has these unique bonding properties, which relays this huge variety of structures. The figure, or the multitude of figures I've put up here for you, are kind of showing some important biological carbon-based structures. I've shown you methane before. We have glycogen, which is an important polysaccharide we'll talk about today. Propane is just an interesting organic molecule. So carbon forms lots of different types of structures that have different characteristics. There's no other element that can form as many large, complex, and unique molecules as carbon, so it really is it's super important, very foundational. These chain and ring structures are the backbone of biological molecules. And molecules that are carbon-based are what we refer to as organic molecules. Now, a key piece to this is typically it has to be carbon bonded to hydrogen If you see carbon bonded to other things, that doesn't necessarily make them organic molecules. It's those carbon-hydrogen bonds that are key. If you take organic chemistry, that's the idea. You would be studying organic molecules, so those carbon-based structures. And then inorganic molecules are those that do not have those carbon-hydrogen bonds. So, in many ways, the diversity that we can observe on the planet is in part due to the diversity of molecules that carbon can form. We also know there's a lot of fossilized remains, like fossil fuels are also made of carbon molecules. So kind of a good thing to keep in mind as we kind of think about carbon's importance. This figure is a little bit basic, but it's just showing some different types of organic and inorganic molecules. Some key differences here. So for the inorganic, we're seeing carbon dioxide, which would be CO2. It would be carbon in that molecule, but again, it doesn't have those hydrogen bonds, or it doesn't have those hydrogens bonded to the carbon. So that's why it's inorganic. Diamond is also made out of carbon, but it's just carbon arranged into a specific form, which we call an allotrope. So again, that's not an organic molecule. Questions here? Sometimes organic molecules are also classified by having carbon-carbon bonds. So if you come across that, that can also be a distinction. We have a huge diversity of carbon-based molecules, and some of those molecules are very similar, as in they will have the same chemical formula, but then structural differences, and that's what we call an isomer. We have two different types of isomers. There's structural isomers, which is where we have covalent bonds located in different places. This example up on the screen here, we have pentane and then this 2-methylbutane. We can see with pentane, the carbons are all bonded together in this linear structure, whereas with the 2-methylbutane, 2 is referring to where that carbon is bonded off the chain. So we can see they have the same chemical formula, the same number of carbons, the same number of hydrogens, but what makes these different molecules is the location of those bonds. So these are structural isomers. We also have geometric isomers, which relates to the position of double covalent bonds. Now, double covalent bonds are relatively inflexible, especially considering like single bonds would be more flexible. With these geometric or cis-trans isomers, the position of the double bond is the same, but we tend to see altered spatial arrangements. So the molecules that are attached to the carbon or the atoms that are attached to the carbon, those are in different positions. Now, differences in the subtle shape of molecules can greatly influence biological activity. For example, butane and isobutane are isomers, and butane is used as lighter fuel. Isobutane is a refrigerant and a propellant. So we see different characteristics even within things that have the same number of carbons and hydrogens. Now, there's one carbon molecule, I'm trying to remember its name. It's C20H42, and it has something like 366,000 different isomers from just one like chemical formula. So there's tons of different isomers out there, and the more carbon-hydrogen atoms you have in a molecule, the more potential there is for isomers. Questions here? Okay. Another type of isomers are these interesting ones called enantiomers. They are isomers that are essentially mirror images. When we think about these, a helpful way to think about it is like a left-handed and a right-handed version of a molecule. Now, they tend to have pharmaceutically different purposes, and a lot of times these enantiomers have very different reactions physiologically. Most of the time, only one is going to be biologically active. And biologically active means there's a physiological effect. So the body is going to be affected by one of these enantiomers. As I was mentioning that pharmaceutical relevance, if one is active biologically and one is not, it's important to use the right form. And then this bottom table is kind of showing effective and ineffective enantiomers on two common drugs. So one of the ibuprofen enantiomers is 100 times more effective than the other. So depending on what you're wanting, that could be important. And then albuterol is commonly used in asthma patients. And then same deal, we have one that's effective and one that's less so. Whoa. Now, there is another enantiomer that is similar for methamphetamine. Obviously, that is a potentially psychoactive and addictive central nervous system stimulant. And then the other enantiomer is actually used for over-the-counter vapor inhalers. So some big differences there. The last thing I didn't really mention, this figure is showing the left and the right hand, but a good way to remember that it's important with those structural differences is like you can't put a left-handed glove on your right hand. So like the location of your thumb matters a lot when putting on gloves. That's the same deal with these. Questions here? Okay. So we're going to get into the macromolecules here. So all of that was kind of talking about the diversity of carbon and the importance of molecular shape. Now, macromolecules are large organic molecules built of smaller molecules. I just said the word molecule like 15 times, but remind me, what does that mean? What is a molecule? Yeah, heck yeah, absolutely. Groups of atoms bonded together. It could be any more than two. It could be lots. It could be few. So yes, we have organic molecules built of other smaller molecules. So basically lots of atoms bonded together. Macromolecules are comprised of something called monomers. And if you break that word down into what it means from its like Greek or Latin roots, mono refers to one and mer refers to part. So this is like one part. And macromolecules comprise almost the entirety of living organisms. So basically all biological organisms rely and are constructed of these macromolecules. And we have four main types: carbohydrates, lipids, proteins, and nucleic acids. I have an asterisk here on lipids because they're not technically macromolecules. Now, every biology textbook that you come across is going to talk about them as macromolecules. It's going to kind of consider them as biologically critical molecules, but they don't hit our definition because they're comprised of like slightly different parts. So it's just like a technical variation here. But they are generally large molecules that are super important, so they do kind of fit. Now, most macromolecules are made of polymers. Again, the exception here is lipids, so that bullet point at the bottom, not all macromolecules are polymers. I'm talking about lipids in that instance. Carbohydrates, proteins, and nucleic acids are all polymers of monomers. There's probably a lot of vocab words you might not have heard before, and that's okay, but I do want to make sure that you're kind of clear, at least for now, and then can kind of review what these things mean later. So again, I have this word here in bold. Monomers are these small molecule subunits, and monomers combine together to form polymers, and polymers are macromolecules. I love this figure. Love is a bit dramatic. I like this figure because it's representing monomers as beads and polymers as the necklace. This is kind of a helpful visualization. In this scenario, we could think of the string as like dehydration synthesis reactions hooking those monomers together. So beads are like the monomers and the necklace is the polymer. Below the bead necklace situation, we have a real-life biological example of monomers and polymers. Here is a glucose molecule, and then the polymer made up of lots of different glucose molecules hooked together is glycogen, which is a very important molecule for storing energy in humans and other animals. Same kind of idea here. This chart I honestly include because I think it could potentially be a helpful study tool for you all. This is showing the monomers and then the polymers or the macromolecules. So on the right side, we have in lighter blue, we have what these specific important macromolecules are made out of. So today we're going to be focusing on those monosaccharides and then carbohydrates. Now, the process of forming these macromolecules is generally by polymerization, which is how those monomers are assembled. And polymerization occurs through dehydration synthesis reactions. Now, dehydration synthesis is when two molecules are combined covalently, so I italicize this, they're combined via covalent bonds to make a larger molecule and a water molecule is released. So that makes sense when we think about the word dehydration. You don't have a lot of water, you're losing water. So dehydration synthesis, you have to lose a water to hook two molecules together. That's what we have here. Now, this process is facilitated in large part by enzymes. I have a whole lecture devoted to enzymes because they're so important and honestly really interesting. But for today, it's valuable to know that enzymes are just important proteins with specific purposes. So there's other proteins that help this reaction occur. And this image at the bottom, this illustration, is showing two glucose monomers being hooked together to form a maltose disaccharide. The last thing to note here, a hydroxyl group and a hydrogen from each of those monomers are being shown here. This is where the water comes from for the dehydration synthesis reaction. That's where the water is coming from. It's actually being taken off of those monomers. Now, this process happens each time. So for every monomer that's added to this polymer chain, there's going to be one water molecule lost. So each time you lose a water. Questions here? If you build something up, you can also break something down. The opposite of building molecules via dehydration synthesis is hydrolysis. I'm also going to break this word down. Hydro refers to water, and lysis refers to breaking. So this is breaking with water. Okay. Now, in hydrolysis, the bonds between our monomer subunits are broken up when water is added, which is what we can kind of see here. Now, I have a question on here for you. Why would it be important for organisms to be able to break down large molecules? Why would that be valuable or important to us? Food, yeah, definitely. So we eat food, we know it gives us energy, but the way that actually happens is those large molecules get broken down, mostly via hydrolysis, into their smaller subparts, and then our bodies can actually use those small subparts to form other molecules that we need. So yeah, definitely important for being able to process nutrition. Now, most of the time, macromolecules are way, way, way too big to be able to enter like the blood system, the bloodstream, in their large form. So that's also why they have to be broken down. That process is also aided by enzymes. Pretty much everything that happens is enzymes are involved. They're very important. So on the previous slide, I was showing you carbohydrates. This is actually a protein or a polypeptide, and then it's amino acids. So that might not mean anything to you today, but in the future, if you go back and you're like listening to this lecture, maybe that'll mean something in the future. As I was saying, dehydration synthesis and hydrolysis require enzymes. They also require and release energy. So dehydration synthesis takes energy. If you're going to build a house, you would need to get some energy to do that, right? Like you would need to be energized to do something, build something. And then hydrolysis releases energy. If you think about our human example, when you eat food, you get energy and the food is going to be broken down, which is what gives you the energy. That's definitely an oversimplification, but I think it can be useful in kind of remembering this process. So I put little stars on these figures with the energy just to remind you that energy is either going to be required or released. Okay, I have a question for you. The electrons in the molecule shown below are being shared unequally. What type of bond is being displayed? Think about this. This is a break question. If you want, you can chat with it. Chat with someone about it. You don't have to, though. What's your break? you have the answer? Yep, we're definitely seeing polar bonds, polar covalent bonds. Yep, absolutely. So in looking at this figure, I think last time I mentioned the delta symbols, the little squiggly dudes with the plus and the minus. So those are delta, lowercase delta, and this is just saying the relative charge. So we can kind of see in this figure, we have a relative positive charge with these hydrogen atoms, and then we have this relatively negative charge in this nitrogen. So that is indicative to us of that unequal sharing. These arrows also represent the direction of attraction. So we can see the hydrogen atoms are being kind of pulled towards the nucleus or towards that nitrogen. Another key to answering this question, I tell you that they're being shared unequally. That's kind of our definition of a polar covalent bond. Covalent bonds involve electron sharing, and then the unequal sharing is the polarity. Does that make sense to everybody, how we got that? If I said they're being shared equally, what type of bond would it be? Nonpolar covalent. Yep, cool. I have another one. This one hopefully should be easier. What are we seeing here? What's going on? Hydrogen bonds. Yes, lovely hydrogen bonds. I could have added more molecules and more hydrogen bonds because I made this figure, but this is, yeah, these little dotted lines are hydrogen bonds. Cool. We're going to keep going. Today, carbohydrates. We love carbohydrates. They're the most abundant organic molecules on the planet, specifically cellulose, which is a large polysaccharide molecule that's common in plants. So there's lots and lots and lots of carbohydrates all over the earth, which is great because they're super helpful to us. And I personally think they're delicious. Which brings me to my question, when you hear the word carbohydrate, what kind of comes to your mind? And I'm actually interested in this question. Like when I'm like carbohydrates, what do you think of? Sugar, grains, sure. Starch, yeah, that's a great one. Corn, yeah, corn is a great carbohydrate. Anything else? Spaghetti, yep, spaghetti, other pasta, yeah. Surprised no one said bread. I usually hear bread when I talk about carbohydrates. But yeah, a lot of times when someone talks about carbohydrates or when we hear this word in like casual conversation, there's a lot of dietary associations mostly with this. But when we break this word down and kind of focus on what it actually means, it just is a combination of carbon, hydrogen, and oxygen in a specific ratio. So we see this ratio of 1 carbon to 2 hydrogens to 1 oxygen in all carbohydrates. And I do have a little break question later where I'm going to ask you to use this ratio. So keep this in your brain or be able to refer back to it later. Now, in your travels, you might read some words that sometimes you don't recognize, but generally, if they end in "-ose, they're carbohydrates. We can think of common carbohydrates like sucrose, glucose, lactose, maltose. They're all ending in that OSE. There are exceptions. Glyceraldehyde, for example, is an important carbohydrate involved in cellular respiration. That doesn't end in "-ose, but a lot of the time they end in "-ose, which can help you identify them. We see different forms of carbohydrates. Sometimes we see the monosaccharides. Sometimes we see polymers of monosaccharide subunits, which would be larger carbohydrates. And a key feature of carbohydrates is the fact that they're soluble in water. Now, soluble means that they could be broken down in water or dissolved in water. And here, this is kind of a sketchy looking slide, but it's just kind of an example. Most common monosaccharides, so the small bits that make up larger carbohydrates, tend to be 3, 5, and 6 carbon molecules. So these are very common, very important monomers or monosaccharides. So ribose and deoxyribose are sugars that comprise DNA and RNA, so the genetic material of cells in all living organisms. Glucose, very important energy molecule, and then fructose and galactose, same kind of deal. That glyceraldehyde molecule I mentioned for cellular respiration is kind of funky looking. This is a chain, but we see lots of important ring and chain structures here. Okay. We have three types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Now, I've done a lot of breaking the words down, which I think is helpful for me to remember what they mean. If that's helpful for you, great. If not, I apologize for doing it so much. Monosaccharides, again, mono refers to one, and then saccharide refers to sweet. So the single sweet, then di refers to two, and poly refers to many. So this is like how many monomers make up this carbohydrate. So monosaccharides are just one, disaccharides are two, and polysaccharides are anything more than two. So if we have multiple subunits hooked together that are our glucose monomers, for example, that would be a polysaccharide. Now, monosaccharides are the main fuel molecule for living organisms. Up here, I've put glucose both in its chain and its ring structure because glucose is required for cellular respiration. It's produced in photosynthesis. It's a really common monomer. It's also the main fuel molecule. If you were admitted to a hospital, for example, or if there's a really sick patient, sometimes they'll actually directly inject these types of monomers or monosaccharides into the blood because it supplies an immediate energy source. So carbohydrates are really important sources of energy for living organisms. That can be helpful for tissue repair or more immediate recovery. I have a question up here. Two monosaccharides form disaccharides. And how are they going to be joined? What is the process called? I talked about it today. Dehydration synthesis. Yeah. The specific bonds between these monosaccharides are called glycosidic bonds. That's just a specific type of bond. Common disaccharides include maltose, sucrose, and lactose. And then I have some bullets here about what those are or how you might kind of like encounter them. Maltose is in germinating seeds, so when they're under the ground, it's a sugar that fuels their ability to break up through the soil. It's also important in the brewing industry, so like malt sugar is something that's in Whoppers. If you've ever had those like malted chocolate balls, those are malt sugar. It's also used in like malted whiskey, so it's a super important sugar in the industry. Sucrose is found in leafy plants. Some plants have a lot more sucrose than others. Things like beets and sugarcane have lots of sucrose, and those are actually used to make table sugar. That sucrose is extracted and then crystallized in plants, and that's how we get table sugar. And when you bite into a piece of fruit and it's sweet because of sucrose, typically. Finally, lactose is that primary sugar in milk that some people do not have the proper digestive enzymes to break apart into its subunits. So if you're lactose intolerant, it means you cannot digest this lactose sugar. Here's that challenge problem I promised. I want you to start here, write the formula for a monosaccharide that has 3 carbon atoms. Once you're ready to talk about the answer, let me know. Or if you want a hint, we could do that instead. Yes, this is exactly that. Is this what you all got? Did you get how we get this? We have our ratio, 1 carbon to 2 oxygen, to 2 hydrogen to 1 oxygen. And then I said, okay, you have 3 carbons, so we just have to kind of scale that ratio accordingly. Does that make sense? Hopefully. Okay, this second bit is a little more challenging, and I think I actually have the answer on the PowerPoint. So don't look at it yet. I want you to think about it in your brain. Lactose is the disaccharide sugar in milk. It's formed from galactose and glucose. The chemical formula for both these monosaccharides is C6H12O6, and that's true. This is all true. I want you to tell me, what is the formula for lactose? So lactose is a disaccharide, and it's formed via galactose and glucose combining to form a disaccharide. So think about this process. How would this occur? And then think about the formula. C12H24O12. Does anyone agree? Yeah, so we have to incorporate that water molecule. That's kind of the trick here. Yeah, so we do have those two monosaccharides combining. Now, the reason it isn't C12H24O12 is because we lose that water molecule. So this is kind of tricky. I probably wouldn't ask you something like this on an exam. It's just a good exercise. But does everyone understand how I got this answer here? You said no? Okay, so this is, like I said, totally tricky. So lactose is our disaccharide, which means it's combined of two monomer subunits hooked together. Now, to answer this question, you have to know that the process that hooks them together is dehydration synthesis. And every time we hook two monomers together, we lose one water. And that also could honestly be a reason why this could be challenging. Our formula for water would be H2O. So we have two oxygens and one water lost for every two hydrogens and one oxygen, lost for every one water. So this is one water. So if we were going to think about this problem, we have our galactose plus our glucose. And they have that same chemical formula, C6H12O6. So we basically multiply that by itself. So we multiply this by 2. And then we would get C12H24O12. But we have to incorporate losing 2 hydrogen and 1 oxygen. So that is why we get C12H22O11, because we lost our water. Does that help? A little bit. If you want more, I can also explain kind of one-on-one in a different circumstance if that's helpful. Yes. Yep. So if I said, okay, let's hook on another glucose, we'd have to consider another water molecule being lost. Yeah, absolutely. Great question. Any others? Okay. I have this kind of written out here for you, the plus sign and then losing the little water. Again, if you want to talk about this more during office hours or over email or anything, I'm happy to try to explain it in the way that makes the most sense to you all. Now, polysaccharides are these large carbohydrate molecules. Mostly their functions are as storage molecules or structural compounds. So storage and structure. When you're talking about carbohydrates, y'all were mentioning starch, which is great. This is made of long chains of glucose monomers. This is generally the storage molecule in plants. So a lot of people say, oh, you're eating like a starchy vegetable, like a potato or a corn, whatever. Yeah, most plants tend to have a fair bit of starch. Now, animals have enzymes, digestive enzymes that are necessary to break starch down. So we can digest the sugar and use it for energy. We can harvest glucose from starch, essentially. Glycogen is the storage polysaccharide for animals. So starch in plants, glycogen in animals. And if needed, humans are able to actually break down glycogen into glucose. So glucose is that immediately usable form of energy. It's small, our cells can use it. It's very helpful, very useful. Glycogen is a big branched structure. So glycogen has to get broken down, but we can also break that down. Cellulose is a glucose polymer. This is mostly a structural molecule. Cellulose is very common in plants. Now, animals do not have the capacity to digest cellulose. We can't digest it. We can't break it down. We do eat a lot of cellulose, and dietary recommendations are to ingest things like vegetables and plants because the insoluble fiber is valuable to our digestive system, but we can't actually break those molecules into their smaller monomer parts. It mostly provides insoluble dietary fiber. Finally, chitin is a really interesting structural polysaccharide. This is common in invertebrates. So invertebrates are organisms that don't have a backbone or a spine. So we're vertebrates. So invertebrates are things like bugs and crabs are actually invertebrates. Chitin is also important to fungi. So I have some examples here of all these different types of polysaccharides. I love cicadas. Some people hate them because they like leave these little crusts, they're exoskeletons on trees, but these are pretty much entirely composed out of chitin, which is that structural polysaccharide. Our starches, of course, getting some of those dietary associations. This figure at the bottom is showing the branched structure of glycogen. Now, glycogen tends to be stored in places like muscles. It's stored in other organs as well. So it's a super important molecule for humans. And if you work out a lot or use a lot of energy and you don't replace your glycogen stores, you can get fatigued over time. So that's why it's important to eat carbohydrates if you're kind of exerting yourself physically. Liver cells also have it. Finally, I have some cotton bath towels here for an example of cellulose. If you've ever used like cotton or linen fabrics, they're pretty strong and they're fairly water absorbent because they're made of cellulose. In part, they're made of cellulose. And we know that polysaccharides or carbohydrates are water soluble, so they like water, they absorb the water, and that's why bath towels sometimes work. So just some examples here. It is also valuable to spend a little bit of time talking about the relationship between like carbohydrates and nutrition. As I was mentioning with our glycogen stores, it is important for organisms, humans for sure, other animals as well, to have carbohydrates in their diet. Now, this image here is of lots of different sources of carbohydrates that are in like a healthy diet. So things like fruits and vegetables. Carbohydrates provide instant energy. If you're going on a run, you want to start with some carbohydrate. Tends to get in your bloodstream fast and provide quick boosts of energy. Some carbs have insoluble and soluble parts. So that insoluble, as I previously mentioned, provides fiber. And then I laid out why that would be helpful here. Adds bulk to waste products, removes cholesterol, and can help regulate blood glucose. So help maintains just important vitals, basically. And then the last slide here is kind of interesting. A lot of people kind of talk about the problems with eating lots of sugar, the problems with eating lots of carbs, and it is valuable to kind of use our critical thinking sometimes here. So the recommended dietary intake of sugar for men and women and men respectively, 24 to 36 grams, is actually not very much. It's like 6 to 9 teaspoons. 71.4% of US adults consume more than the recommended amount of added sugars, which I definitely do that sometimes. It's really easy to because it's in a lot of processed foods. There is some correlation between high added sugar and some health problems, things like high blood pressure, high cholesterol, diabetes, cardiovascular disease. The key here is with added sugar. So that would be like adding sucrose, adding honey, adding things that aren't naturally present. For example, in things like fruit, there are those additional vitamins and fibers that don't produce the same health outcomes. Now, the nutrition label I have up here is where that critical thinking comes in. This is kind of interesting. So I pulled this exactly off of a can of root beer. So you'll notice I have an arrow pointing to the includes, what does that say, 41 grams of added sugar. Okay, and it's saying that's 81% your daily value of sugar, which if we look at kind of some of the dietary guidelines, that might not actually be accurate. So this is just a good reminder to think about kind of some of the things you read. So that's all I have for today. On Monday, we'll talk about proteins, lipids, and nucleic acids. Have a good weekend.